Hi, my name is Eloi Schmar. I'm a PhD student and a member of Monolith Scalis Lab. I work in transcriptomics, especially looking into dissection of health and diseases. So a recent project that I found myself to be very passionate about is xenotransplantation. So there is a shortage of organs over the world for transplant and for patients who need a transplant. And recently with gene editing abilities, there has been an interest into trying to use animal organs, especially pig organs, for human transplants. And one of the current research that is going on about that is trying to test these into human dissident subjects and understand the immune response and try to adapt the different immunosuppressant drugs in order to make this possible. And one way to do that is to look at the tissue of the subject, the dissident, both their blood and as well as the transplant, and try to look into the cellular response and the processes involved, and basically looking into the state of the tissue and the cells within them, and to try to then improve the way that we do these transplantations. I'm working on the side of the data analysis, so a lot of samples have been collected and have been processed, especially a lot of sequencing has been done, trying to understand the different levels of RNA and DNA and other molecules. And my role in this project is to analyze that data and make sense of it. So as I'm a bioinformatician, I have to look into different ways to process the data, to work as a data analyst, but also as a biologist to interpret the different changes, the immune changes that are happening in these samples. So on the more technical side, one part that is actually for me quite exciting, because I really like all of that informatic work, is when the data comes in, what it looks like and what are the first steps. And the first issue is that these are usually very big data sets in terms of size. So go into several hundred of gigabytes usually for a data set. And obviously we process that into our high power compute servers, high capacity compute servers. And the first step is to basically get the raw data into a sequence format. So there we get a lot of the data is sequencing. So there we get sort of text files that contain the list of nucleotides, A, T, C, G, even though it's RNA, so it should be AU, CG. They are huge text files. Simply, that's the data we get out of the sequencing machine in simplest term. And then there are different programs that we run to align that to the genomes. And then from that on, we get what we call processed data, which is, in our case, in the example for single-cell RNA-seq, which was one of the biggest data sets we go with, is simply a matrix with rows and columns and numbers in the middle, and the rows are the cells and the columns are the genes. So for each cell, we know what is the quantity of each gene in RNA levels. So this is transcriptome, is simply a view for all genes. We have 20,000 genes in the human genome. So that can be huge matrices. We can get several thousands or tens of thousands of cells. And this is the basic process data that we work with, on which we do different transformations and analyze this to understand better. So on the technical side, I work a lot on Python to analyze that, but before that, it's a lot of bash scripts and different programs that exist to be able to process this. This is actually a non-trivial part of the work as an informatician, but it's also for me one of a very satisfying and interesting work. There's all the technical aspects of processing all of that, and you get access to very powerful computers with a lot of storage. And it's, for me, I find that very cool, actually.